At the moment of somatic death, the body initiates an intricate, highly structured transfer of energy and matter. Over the first 12 hours, the complex biochemical mechanisms that maintain the body's internal environment shut down, submitting to the laws of thermodynamics. Macroscopic changes in the cadaver are driven by two specific microscopic events. The first is the total depletion of intracellular ATP within the muscle fibers. The second is the autolysis of the mucosal tight junctions inside the gastrointestinal tract. To understand the progression of rigor mortis and early putrefaction, we must map this sequential cellular shutdown as it unfolds over the first 12 hours. By the second hour postmortem, cellular respiration has ceased. Without respiration to generate new energy, the body's remaining intracellular ATP reserves are exhausted. This energy depletion manifests macroscopically as rigor mortis, beginning in small muscle groups like the eyelids, jaw, and neck. Within the muscle fiber, the sliding filament model shows how contraction works during life. Calcium binds to troponin, exposing binding sites on the actin filaments. The myosin heads then attach to form a crossbridge, pulling the actin filaments to contract the muscle. Muscle relaxation is an active process that requires a constant input of energy. Specifically, the detachment of the myosin head from the actin filament requires the binding of a new molecule of ATP. When ATP levels reach zero, the crossbridge cycle freezes mid-stroke. The myosin heads remain permanently locked to the actin filaments. This microscopic locking mechanism creates a rigid state that progresses downward through the extremities. This rigidity holds for 24 to 36 hours until the physical degradation of the structural myosin proteins causes the muscles to relax. Rigor mortis is the macroscopic manifestation of an exhausted microscopic energy reserve, holding the system in place until structural decay begins. While the muscles lock, a different structural collapse occurs in the gastrointestinal tract. The gut houses trillions of microorganisms, an ecosystem that outnumbers the host's own cells. During life, this population is contained by a continuous physical barrier of enterocytes. Tight junctions seal the spaces between these cells, preventing bacteria from escaping the lumen. This is supported by an aggressive innate immune system, with macrophages and neutrophils patrolling the barrier. By hour 6 postmortem, oxygen deprivation has killed the immune cells, removing active resistance. Simultaneously, cellular autolysis causes the tight junctions to dissolve, and the intestinal barrier fails. With the immune system dead and the junctions dissolved, the host can no longer contain its own microbiome. Starved of their normal food sources, the gut bacteria begin to migrate. Anaerobic species, such as Bacteroides and Clostridium, initiate a mass migration known as bacterial translocation. The bacteria break through the disintegrating mucosal wall and flood into the submucosa. They gain entry to the lymphatic ducts and the stagnant blood vessels of the mesenteric system. These stagnant vessels serve as a nutrient-rich highway, allowing microbes to spread to the liver, spleen, heart, and brain. The bacteria feed on the amino acids and sugars liberated by the host's cellular autolysis. This transition marks the end of host-driven autolysis and the beginning of microbial putrefaction. Utilizing these biological remains, the microbes multiply exponentially. Their metabolism produces off-gassing byproducts, including hydrogen sulfide, methane, putrescine, and cadaverine. Putrefaction is an aggressive, exponential colonization by an ecosystem utilizing the host's remaining infrastructure. At hour 12, the cadaver is fully engaged in the process of putrefaction. The progression of these 12 hours illustrates the law of conservation of mass and energy within a biological system. From a molecular perspective, the biological matter of the body is being systematically reorganized. The complex structural proteins, essential lipids, and nucleotides that defined the body are completely dismantled by autolysis. This matter is repurposed, serving as the high-energy substrate required for microbial life to flourish. The body does not simply vanish. Its components are integrated into a vast, emerging ecosystem. Biological death is not an endpoint, but the thermodynamic ignition required for new life to begin.